tropomyosin, the strand-like protein, off of the myosin binding sites on actin. So calcium binding to troponin exposes the myosin binding sites on the different globular actins. Because you have to remember that troponin is attached to both tropomyosin and to actin. And that when calcium comes along and binds to it, troponin changes its shape. And remember, it's bound to tropomyosin. And as it changes its shape, what it does is it pushes tropomyosin off of the myosin binding sites on actin. And that allows for myosin and actin to be able to interact with each other, which is called a contraction, which is what generates force inside of our muscle fibers. So, I said this a lot when we were discussing metabolism, but I said when a muscle fiber contracts, we know that calcium rises inside of it. And the more that a muscle fiber contracts, the more calcium rises. And I told you one thing that calcium can do is it can assist with breaking glycogen back down into glucose 6-phosphate. But the real most important reason that calcium elevates inside of our muscle fibers during contractions is for this. We need calcium to elevate so that it can bind to troponin, unwind tropomyosin, and allow for myosin and actin to interact with each other. And then we can have one of these contractions. Okay, so that's the first thing that we need. It's calcium in order for a contraction to happen. We'll come back to that in a little bit, but for now, let's focus on the structure of myosin. What about the myosin filament? The myosin filament is the thick filament that comes off of the M line. How is it structured? You've probably noticed by now, and probably have seen in anatomy, probably talked about it in lab to some degree, there are all of these myosin heads located on the myosin filament. And where do these myosin heads come from? It looks like you've got this filament and then all these little heads that come off of the filament. And that's really not how it's organized anatomically. What you have with the myosin filament is actually very similar to what you have with the actin filament. You know how the actin filament is really made up of a whole bunch of globular actins, G-actins? Well, the myosin filament is actually made up of a whole bunch of what are called little myosin proteins. So let's take a look at one of those myosin proteins. And then I think this image will make more sense. This is one myosin protein. A myosin protein, first of all, has what's called a tail. There are two what are called heavy chains that wrap around each other in a double helix, and they form the tail. I wouldn't ask you about the heavy chains on the exam, but they're two separate proteins that actually wrap around each other and form this tail portion of the myosin protein. And then what happens is at the end, the two heavy chains that form the tail diverge from one another, and each forms a head. So each myosin protein has a tail and two heads. And each head has a site on it that's called an ATPase site. That's actually an enzyme that breaks down ATP. And then there's another site on each head called the actin binding site. The actin binding site on the myosin protein heads is what's going to bind to the myosin binding site on the globular actins. But again, we'll get to that in a little bit. Let's just focus for the moment on kind of the structural anatomy here. Okay, so each myosin protein has a tail and two heads. And let's just focus on the fact that each head basically combined ATP and break it down. All right. A myosin filament is just made up of hundreds and thousands of these myosin proteins. So now let's take a closer look at that image here. Look at this. What you're actually looking at with the myosin filament is just thousands of myosin proteins all collectively together. Again, if you look closely, you can see all the tails. Here's a tail, here's a tail, right? There's a tail, there's a tail, there's a tail, there's a tail, there's a tail. All of these little striations that you're seeing are tails. They're myosin protein tails. And you may notice that the tails of the myosin proteins lie parallel to the actin filament. But what about the heads? The heads exist on an angle, which you can clearly see in the image. And that angle causes the heads to exist slightly perpendicular to actin. So the heads are orientated in a way that allows for the heads to latch on and bind to actin. So now we can kind of collectively define the myosin filament. The myosin filament is again actually made up of thousands of myosin proteins. Each myosin protein has a tail and two heads. The tails of the myosin proteins lie parallel to actin, and the heads of the myosin proteins are on a bit of an angle. They're cocked upwards or downwards, depending upon what side of the myosin filament we're looking at. And they're then orientated in a way where they are slightly perpendicular, the heads to actin. All right, so that is the sort of basic organization of the sarcomere that we're going to look at. Okay? There's actually a lot more to talk about with sarcomeres, but for the moment, that is all we're going to do. What I really wanted to try to accomplish there was for you to see a little bit more detailed anatomy as to what the actin filament actually is and what the myosin filament actually is. So now let's actually take a look at the way that myosin and actin interact with one another during a contraction. This is what's referred to as the cross-bridge cycle. The cross-bridge cycle is the way in which the myosin heads interact with actin within the sarcomere. We're going to start here. This is what's called the rigor or Riger state. We heard of rigor mortis before. That's where this comes from. This is where the myosin head is actually bound to actin. The myosin head, if you take a look here, the myosin head is bound to a myosin binding site on a specific globular actin. And force is actually being generated here. But the myosin head is stuck to actin. And what the myosin head needs to release from actin is ATP. So what will happen is ATP will come along and it will bind to the myosin head. Remember, there's an ATPase site on the myosin head, meaning the myosin head can bind ATP, and that's what happens here. ATP binds to the myosin head, and when ATP binds to the myosin head, that's what allows for the myosin head to release from actin. Oh, yeah, go ahead.
I was just wondering, is that why during rigor mortis, someone's muscles are kind of locked up because there's no more ATP to release the myosin heads from actin?
Yeah, and it also doesn't start right away. So if a human or another animal dies initially, you don't have, this is one of the ways that you can identify how long ago the person died. Initially, there is no rigor mortis. It sets in after a certain amount of time. And what you actually need is for the cells to begin to die, so that calcium begins to flood in to the muscle fibers first. And then when calcium floods in, it binds to the troponin, exposes the myosin binding sites on actin, and then the myosin heads bind to actin, but there's no ATP in the cell anymore because the cell is dead, it's not producing ATP, it's not receiving oxygen. And without the ATP, the myosin head is just permanently stuck to actin, and then the muscles are contracted and stiff. So, yeah, good question. Okay, so the myosin head is bound to actin, and if it wants to release from actin, it needs ATP. This is something that we know for certain. Now, as the myosin head releases from actin, what it does is it consumes the ATP. It hydrolyzes the ATP. And what we actually say is that the myosin head becomes phosphorylated. The reason that we say this is because the ADP and inorganic phosphate don't leave the myosin head, they stay within the myosin head. So the myosin head retains that phosphate group as it moves backwards. So in essence, the myosin head has been phosphorylated. It phosphorylates itself. Then it cocks back upwards and it grabs on to a new myosin binding site on a different globular actin. Although, during something like an isometric contraction, it could potentially grab onto the same one. That's a little bit more of an advanced conversation. We can come back to that in a moment. So, anyways, the myosin head moves upwards, it grabs on to usually a new globular actin. And the next thing that happens is what's referred to as the power stroke. The myosin head becomes dephosphorylated. The phosphate group exits. So now the myosin head loses that phosphate group. And the loss of the phosphate group causes the myosin head to swivel towards the M line. So if this were a Z disc here, and this were the M line, when the myosin head swivels or cycles, it pulls the actin filament towards the M line. And then, after the myosin head has swiveled towards the M line and pulled the actin filament towards the M line, the last thing that happens is the ADP gets released, and we've now returned to the rigor state. We have now returned back to where we started. The myosin head is bound to actin, force is being generated, and the myosin head is stuck. If you want the myosin head to release from actin, then you need another molecule of ATP to come along and bind to the myosin head, and that will cause the myosin head to release from actin. As the myosin head releases from actin, it consumes the ATP. It hydrolyzes it. and the myosin head becomes phosphorylated. Then the myosin head moves back upwards, grabs on to a new G-actin, myosin binding site on G-actin, and becomes dephosphorylated. After it grabs onto that new globular actin, the phosphate group leaves, the myosin head becomes dephosphorylated, and you get what's called the power stroke. The myosin head swivels towards the M line again and pulls the actin filament towards the M line. Then ADP is released, and again, you're back to the rigor state. This is the means in which the myosin heads cycle. That whole process, again, is called the cross-bridge cycle. And what the cross-bridge cycle allows for, typically, although this is by no means universal, is shortening of the sarcomere. So as this cycling process occurs, and the myosin heads cycle towards the M line, they progressively pull actin towards the M line, and as actin moves towards the M line, myosin and actin overlap each other more and more. This is not a very great image of this, but that means if actin and myosin overlap each other more, that the sarcomere is shorter. And this is where the term sliding filament theory comes from. It comes from the fact that the filaments, actin and myosin, particularly actin, slide over one another. What really happens is the actin filaments slide over myosin as myosin pulls the actin filaments towards the M line as the myosin heads cycle. This was discovered in the 40s and 50s by a famous scientist, A.F. Huxley, in the United Kingdom, and it was done with amphibian, it's a frog muscle basically, and you would electrically stimulate the muscle, and you would notice that essentially these different bands, which were actually myosin and actin, would slide over each other when the muscle received electricity. Because the muscle, as you can see, needs electricity to contract. So that's where the whole idea of the sliding filament theory comes from. It's the physical observation made by scientists that these filaments were sliding over each other during a contraction. And what was actually happening was the myosin heads were cycling, pulling the actin filament towards the M line, and that was causing myosin and actin to overlap each other more. Interestingly, the guy did not win the Nobel Prize for that, but rather he discovered action potentials. So really prolific and important scientist. But anyways, he did that with squid tentacles. So what happens during one of these contractions? What do you need? You need really two things. You need three things, which also is electricity, which we'll get to next. If you want a muscle fiber to contract, you need calcium to elevate inside of the muscle fiber. Because if calcium doesn't elevate, myosin and actin can't interact. So you're not going to get a contraction. You need calcium to elevate so it can bind to troponin, which will expose the myosin binding sites on actin. Then myosin and actin can interact with each other. But if you also want the contraction to continue, you need ATP. And we spent an entire module talking about how muscle fibers and other cells in the body make ATP. And hopefully you can now see also to a greater degree, the more that you ask a muscle fiber to contract, be it forcefully and frequently, the more ATP it's going to consume. Because the more cycling of the myosin heads you're going to get. And for each time the myosin head cycles, it consumes a molecule of ATP. So the more of these that happen, the greater the ATP consumption, which will influence glycolysis and so forth and so on. Okay, let me finish with this, and then we'll move to the next PowerPoint. Traditionally, and you have to start somewhere, the way that muscle fibers are thought to contract is, well, the muscle fibers shorten when they contract. Although muscle fibers can contract and not shorten, and muscle fibers can actually contract while they lengthen. So it's a bit of an oversimplification, but it's helpful in beginning phases of understanding this to just talk about the shortening part. So how does a muscle fiber shorten? First of all, you have to start the contraction, which is going to be caused by a neuron. So we'll get to that part in a little bit here, but a contraction is happening. If a contraction happens, calcium will elevate inside of the muscle fiber. The muscle fiber already has ATP, all right. What happens during a contraction? Well, all of the myosin heads start cycling and pulling actin towards the end line. And actin then slides over myosin, and the sarcomeres shorten. So during a contraction, the sarcomeres shorten. And what I would ask is, what is a myofibril made up of? Sarcomeres. So if the sarcomeres shorten, and the myofibrils are made up of sarcomeres, literally they're pretty much just made up of nothing but sarcomeres, then the myofibrils have to shorten themselves. Because the myofibrils are structurally nothing other than just a bunch of sarcomeres. So if sarcomeres are shortening, the myofibrils are shortening. And then I would ask, well, what about the whole muscle fiber? What's the muscle fiber made up of? It's 80 to 90% nothing other than myofibrils. The whole interior of a muscle fiber is basically all myofibrils. If the myofibrils are shortening, and the muscle fiber is made up of 80 to 90% myofibrils, then the muscle fiber must be shortening. Because structurally, that's what the muscle fiber is. So this is how muscle fibers shorten. It starts at the level of the sarcomeres. If the sarcomeres shorten, then the myofibrils shorten. If the myofibrils shorten, then the muscle fiber shortens. And then we can end this and say, what is a muscle made up of, like a biceps muscle? The biceps muscle is made up of hundreds of thousands of muscle fibers. Now what if the muscle fibers within the biceps muscle were to shorten? Well, if muscle fibers within the biceps muscle were to shorten, and the biceps muscle is mostly made up of muscle fibers, then the biceps muscle will shorten. And that's how we see muscles shorten during contractions. Specifically what are called concentric contractions. A concentric contraction is when a muscle contracts while it shortens. So if you can contract any muscle and it'll cause the muscle to sort of ball up. What's happening is the muscle is shortening. And now you can think the reason that that whole muscle is shortening during a contraction is actually starting at the level of the individual sarcomeres within all of the different muscle fibers inside of that whole muscle. The sarcomeres within the muscle fibers are shortening, causing the myofibrils to shorten, causing the muscle fibers to shorten, and if all the muscle fibers are shortening, then the whole muscle itself is shortening. And then what that shortening does is it pulls one bone towards another bone. So as the biceps muscle shortens, it pulls the radius towards the scapula, right, and it allows for elbow flexion. So this is how we get human movement. As I said, there are what are called isometric contractions and eccentric contractions, which require a little bit more of a discussion that we don't have time to get into, but this is the kind of basics in which contractions happen and move the body. And let me also say, how is that force, I'm going to say this, where does force come from? This will be important for our next part. I think it's probably pretty clear from the discussion, but force comes from the interaction of myosin and actin. Force comes from contractions. When myosin and actin interact with each other, they generate force. So force generated by a muscle fiber is proportional to the amount of myosin and actin that are interacting with each other. The more myosin heads you have interacting with actin, the more force you'll get inside of the muscle fiber. But how does the force that's generated through the interaction of myosin and actin actually reach the tendon and then the bone, so that the bone can be pulled towards the other bone and cause human movement? Well, in the most basic way possible, let me just say this. I don't expect you to memorize any of this part, but if you're just curious, what happens is, when myosin and actin interact with each other, they generate force, and the force always gets relayed onto the Z discs. And there are little stringy filaments that attach to the Z disc. They're called intermediate filaments. The most common one that's known is desmin. And this intermediate filament is wound all throughout the muscle fiber, and ultimately it attaches to the plasma membrane. And where it attaches to the plasma membrane is onto these little proteins called costomeres. And the costomeres, these little proteins, actually attach to the endomysium, the connective tissue that surrounds the muscle fiber. So this is what would happen, basically. Myosin and actin would interact with each other during a contraction, and they would generate force. The force that myosin and actin generate by interacting with each other gets relayed onto the Z disc, which gets relayed onto the little stringy filament that attaches to the Z disc, which gets relayed onto these proteins attached to the plasma membrane, which then gets relayed onto your endomysium. And if you remember, your endomysium is continuous with all the other connective tissue layers and your tendon. So when you relay force onto the endomysium, you relay force onto the tendon, which relays force onto the bone, which moves the body. Okay. There's a lot of proteins that are involved with this. One of them that's involved with this is what's called dystrophin. You've heard of muscular dystrophy before. That protein is dysfunctional in people that have muscular dystrophy, and basically force can't get relayed from myosin and actin interacting to the endomysium. So the muscle fiber literally just rips itself apart because it can't translate and relay force correctly. And then the muscle fibers die over time because they literally destroy themselves because they can't relay the force correctly. Okay? So, hopefully that's provides a little bit of insight. Again, I'm not going to ask you about how force is relayed on the exam. I do want you to know that force comes from myosin and actin interacting, but you don't need to know the desmin and costomeres and everything else. You can tell by looking at the objectives too, but you don't need to know. This is part 3. We'll get started with this. Probably define the brain. That's probably about as far as I'll get today. What you're looking at here is the cerebrum. The cerebrum is a part of the brain. Most people just think about it as being the brain, but there's actually multiple parts to the brain. The brain is made up of the cerebrum, it's made up of the cerebellum right here, it's made up of the brainstem, you have the diencephalon, so there's more to the brain than just the cerebrum, but all we're looking at is what's called the cerebrum. The biggest part of the brain that everybody thinks about as being the brain. So, right there. If you've done a dissection before in something like anatomy, you can take a brain of essentially any animal, be it a cat, a pig, whatever you use in the lab, or some people might not view that as being lucky, but if you're really interested in anatomy, if you're lucky enough to do an actual human dissection, you can certainly take one of those brains and cross-cut it. Right, this is a lateral view, so like this view right here would be like looking at the left side of my brain right here, my cerebrum, and essentially what you do is you take a knife and you create what's called the coronal or frontal cut, and you cut the brain into a front and a back half. And then, let's just pick a spot, and we'll use the one that they chose, which is right here. So we do a coronal cut of the brain, and then you have to imagine that I come over here, and I pull off the front half, I spin it around, and now you're looking at the inside of it. And that's what you would see. You'd see a distinct outer layer, which is gray, and an inner layer, which is white. So again, you'd see an outer layer of the cerebrum, which is gray, and an inner layer, which is all white. That's called gray and white matter. This exists everywhere throughout the cerebrum. You can cut it at any point. You could cut it here, you could cut it here, 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 here. Makes absolutely no difference. If you cut it at any one of those locations, if you cut it at any one of those locations, you pulled it apart and you looked at the inside of it, you'd see the exact same thing. You'd see an outer layer of gray and an inner layer of white. That outer layer of gray matter is what's called the cerebral cortex. Cerebral means cerebrum, cortex means outer layer. The cerebral cortex is gray. It's gray matter. It looks kind of more brownish in this image, but it's called gray matter. And then you would see that the core of the cerebrum is white. Now, why does it have this orientation, this coloring scheme? Well, particularly if you're looking at the front half of the cerebrum, it's because of this organization. The back half is a little bit different. We're going to talk about the front half, which is more so considered the motor part of the cerebrum, and the back half, although it's a generalization, it's more so considered the sensory part. So if you look at the front half, the reason that you see this organization is because of this. Gray matter is always made up of cell bodies and dendrites of neurons or fully unmyelinated neurons. Neurons that don't have myelin sheath on their axons. That's what gray matter is going to be. Okay, so we can say we've got cell bodies and dendrites that make up the gray matter. In the back half of the brain, it's more so whole neurons that are unmyelinated. And in the front half, it's more so just cell bodies and dendrites of neurons. All right, so where are their axons? Where are these neurons' axons? Well, their axons project into the core of the cerebrum and down the spinal cord. And this is what makes the core white, because these neurons are myelinated. And myelin is fat, and it appears white. Think of butter. So this is how the front half of the cerebral cortex is largely organized. The cell bodies and dendrites all exist up in the cerebral cortex, which is what makes the cerebral cortex in the front half gray. And then the axons, which are myelinated, project into the core of the cerebrum and down the spinal cord. And since the axons are myelinated, that makes the core white. So now let's focus more so on muscle. This is the cerebral cortex again, but what's being done is different parts of the cerebral cortex are being highlighted to denote different functions. So the first thing that you need to do is find what's called the central sulcus. The central sulcus is a groove that separates the front half of the cerebrum from the back half. So find the central sulcus. That separates the front from the back. And now, go right in front of the central sulcus, and you'll find what's called the primary motor cortex. The primary motor cortex is the part of the cerebral cortex, the gray matter, that exists right in front of the central sulcus. Again, find the central sulcus, go right in front of it, you'll find the primary motor cortex. It's a small strip of the cerebral cortex, just a small portion of the cerebral cortex, that controls basically all of the skeletal muscles in the entire human body. This sliver of your cerebrum is what allows for all of your different muscles to contract. And we've known this actually for a really long time, well over 100 years before we understood much about muscle actually, because you could electrically shock someone in different parts of their brain, and it became evident that that caused different muscles to contract. Now we can do this in a much more precise way these days with something that's called TMS, which is transcranial magnetic stimulation. But in the past, there were means of accomplishing this too, which were a little bit more dangerous, but were still used, where you could like precisely shoot electricity into someone's brain. And what was clearly observed is that when you shocked different parts of the cerebrum, muscles wouldn't contract. A person might experience different sensations, but muscles wouldn't contract. And then all of a sudden, when you started putting electrical impulses into this specific part of the brain, which again is like right here, all these different muscles started contracting. So it became evident that this was the part of the brain that controlled the different skeletal muscles in the human body. And now, I mean, there's much easier mechanisms for discovering this with things like functional MRIs, but my whole point is this has been fairly clear to people for a long time. Anyways, how does the primary motor cortex then accomplish controlling all the different skeletal muscles in the human body? We'll talk about that on Friday. Okay?